Chapter 29, Heredity.
Heredity is all about how you receive the characteristics you have. What determines that are DNA segments, otherwise known as your individual genes, which contain the genetic blueprints for proteins. These proteins are mostly enzymes, and their job is to dictate the synthesis of practically all the body's molecules. They are what is responsible for your hair color, sex, blood type, etc.
Assuming there are no issues, the nuclei of all human cells, except gametes, have 23 pairs of chromosomes. Each pair consists of one chromosome from the father and one chromosome from the mother. Two of the chromosomes are sex chromosomes, X and Y. The male sex chromosomes, X and Y, and the female sex chromosomes are X and X. The other 22 pairs, 44 chromosomes total, are autosomes, which guide the expression of most of other traits. There are also the genome, which is the genetic makeup that represents two sets of genetic instructions, one from the egg and one from the sperm.
Sex chromosomes aren't the only things that are paired. Genes are paired as well. Each of us receives two genes, one from each parent, that interact to dictate each trait. Alleles are what we call genes that become matched in the same location of homologous chromosomes. Their job is to code for the same or an alternative form of a given trait. Homozygous is the term used to describe a person when a pair of alleles are the same when controlling a trait. On the other hand, a person can also be heterozygous. This is when the pair of alleles are different. Alleles can also be dominant or recessive. A dominant allele suppresses the expression of its partner. This then causes the allele that is masked to become recessive. Because the dominant allele is stronger, recessive alleles must be presented in double dose if they be expressed. This only accounts for the homozygous condition.
Genotype is your genetic makeup, and phenotype is how that genotype is expressed in the body. Someone being double-jointed is an example of the phenotype produced by a genotype. Not everyone has this condition though, and that is because every person has their own unique genotype and phenotype. This variability is because of three events. 1. Independent assortment of chromosomes. 2. The crossover of homologs. And 3. Random fertilization of eggs by sperm.
During both spermatogenesis and oogenesis, each pair of replicated homologous chromosomes synapses during meiosis I, forming a tetrad. Maternal and paternal chromosomes are randomly distributed to daughter nuclei because change determines how the tetrads line up. Because of this, there is a huge amount of variation amongst the gametes. Additionally, the number of gamete types increases drastically as chromosome number increases. However, when compared to men, the number of different gamete types produced in women is significantly less.
Crossing over and exchange of chromosomal parts during meiosis I results in gamete variation. Genes are said to be linked because they are transmitted as a unit to daughter cells during mitosis. However, chromosomes can break and exchange gene segments with other homologous counterparts during meiosis. This exchange gives preparation to recombat chromosomes which have mixed contributions from each parent. A crossover may also happen where a break occurs between the linked genes. A crossover results in the two of the four chromatids present in the tetrad to end up with a mixed set of alleles. This means that when the chromatids are segregated, each gamete will receive a unique combination of parental genes. And because humans have 23 tetrads, there is a huge variability within the results.
Most human traits are determined by multiple alleles or by the interaction of several gene pairs. Dominant recessive inheritance reflects the interaction of dominant and recessive alleles. A Punnett square can be used as a way to find out, for a single trait, the possible gene combinations that would result from the mating of parents of known genotypes. However, this method is not entirely accurate and predicts only the probability of a certain genotype and phenotype.
Some normal examples of human traits dictated by dominant alleles include dimples, freckles, and a widow's peak. Recessive inheritance is considered more desirable. An example of this is normal vision. However, there are also disorders. A disorder that has to do with dominant inheritance is Huntington's disease. It is a fatal nervous system disease involving degeneration of the basal nuclei. It has a delayed action gene where the disease isn't expressed in an individual until age 40. Additionally, offspring of a parent with this disease have a 50% chance of getting it. A disorder that has to do with recessive inheritance is albinism, which is a lack of skin pigmentation. Recessive genetic disorders are more common than dominant genetic disorders. Also, recessive disorders do not show how, but can be passed on.
Some traits exhibit incomplete dominance. This is when the heterozygote has a phenotype intermediate between those of homozygous dominant and homozygous recessive individuals. An example of this is sickle cell anemia, where the person who has it has lowered blood oxygen levels due to a double dose of the sickling allele. Normally, we inherit only 2 alleles for each gene. However, some genes exhibit more than 2 allele forms, which is called multiple allele inheritance.
Inherited traits determined by genes on the sex chromosomes are said to be sex-linked. The Y, which contains the gene that determines maleness, is much smaller than the X chromosome, which is why there are more X sex-linked traits than there are Y. Also, a gene found only on the X chromosome is said to be X-linked. When a male inherits an X-linked recessive allele, its expression is never masked because there is no corresponding allele on its Y chromosome. Very few females exhibit the X-linked conditions, and X-linked traits are typically passed from mother to son, never father to son. This is because males receive no X chromosome from their father.
Polygene inheritance is where most phenotypes rely on several gene pairs at different locations acting in tandem, which results in continuous or quantitative phenotypic variation between 2 extremes and explains many human characteristics, for example, your skin color, height, metabolic rate, and intelligence.
Environmental factors definitely influence gene expression. For example, drugs or pathogens alter normal gene expression during embryonic development. Phenotypes are environmentally produced phenotypes that mimic conditions that may be caused by genetic mutations.
Our genome is a biochemical system of awesome complexity, with 3 basic levels of controls. The protein coding genes make up the first level and account for less than 2% of the DNA of a human cell. The second layer appears to be the product of abundant RNA-only genes. It is now believed that 80% of the genome serves some purpose beyond simply defining proteins. These small RNA molecules can act directly on DNA, other RNAs, or proteins. They control timing of programmed cell death during development and can also prevent translation of another gene. Mutations in these RNA-only areas have already been linked to several conditions, such as prostate and lung cancer and schizophrenia. Lastly, the third layer of gene controls is epigenetic marks. This continually changing info is stored in the proteins and chemical groups that bind to the DNA and in the way a chromatin is packaged in the cell. Epigenetic marks, or lack of them, predispose a cell for transformation from normal to cancerous, and even slight variations in the marks can lead to terrible human illness.
Not all the genes are in the cell's nucleus. 37 genes are in the mitochondria. Mitochondrial genes are transmitted to the offspring almost exclusively by the mother. There are some disorders, although rare, that are linked to errors in the mitochondrial genes. Degenerative muscle disorders or neurological problems have been associated with this issue.
There are 2 major ways to identify carriers of detrimental genes. These ways are by the use of pedigrees and blood tests. A pedigree traces a genetic trait through several generations and helps predict the future. Simple blood tests are used to screen for the sickling gene in heterozygotes, and more complicated blood chemistry tests and DNA probes can detect the presence of other unexpressed recessive genes.
Fetal testing is used when there is a known risk of genetic disorder. The most common type of fetal testing is amniocentesis, where a wide-bore needle is inserted into the amniotic sac through the abdominal wall. This is done to check the fluid for enzymes and other chemicals that serve as markers for specific diseases. Another type of fetal testing is chorionic villus sampling, or CVS, which suctions off bits of the chorionic villi from the placenta for examination.
Advances in diagnosing human genetic diseases have been followed quickly by new applications of gene therapy to alleviate or even cure the disorders. One way to do this is to infect the defective cells with a virus into which a functional gene has been inserted. This approach has successfully treated some severe combined immunodeficiency disorders.